Alright guys, welcome back to Underground Science, and in this video we're going to be talking about the purine salvage pathway. Specifically though, we'll be talking about the drugs involved, or the drugs that you can use for this pathway specifically. So what drugs inhibit what enzymes in this pathway. Alright, so let's go and get started. So first things first, let's focus on, so we already covered the purine salvage pathway in the previous video, so make sure and go check that out. But first things first, let's go ahead and focus on adenosine deaminase. If we remember, adenosine deaminase, right, took adenosine and converted it into inosine. All right. So if we have drugs that inhibits this enzyme, right, we can't make inosine, and therefore we cannot go ahead and make hypoxanthine and then make uric acid. All right. So if we inhibit adenosine to inosine conversion by inhibiting adenosine deaminase, we'll have a buildup of adenosine, and we'll go over adenosine deaminase deficiency in another video. But for now, let's just know the drugs, or let's just know what drugs inhibit what enzymes in the purine salvage pathway, because that's high yield. All right. So first things first is we have our first drug that inhibits adenosine deaminase, which will lead to you not being able to convert adenosine into inosine. The first drug that we should know is called cladribine. All right, and the other drug that inhibits adenosine deaminase is called pentostatin. All right, and the other drug that inhibits adenosine deaminase is called pentostatin. All right, so those are the drugs that inhibits adenosine deaminase. Okay, the other enzyme we should focus on is xanthine oxidase. Now, there's two drugs that we should know that inhibit xanthine oxidase. All right, so these two drugs inhibit xanthine oxidase. What are these two drugs? The first drug we should know is called allopurinol. All right, and the second drug is called febuxostat or febuxostat. Definitely, totally not pronouncing that right. Febuxostat. All right, so the first drug is allopurinol. Second drug is febuxostat or febuxostat, right? However you pronounce that. But these two drugs inhibit xanthine oxidase. So what does that mean? If you inhibit xanthine oxidase, that's ultimately leading to decreased uric acid production, right? And that's a good thing, especially for people, let's say, for example, who have gout. So you want low uric acid levels because they have hyperuricemia, meaning high uric acid levels. If you give xanthine oxidase inhibitors like allopurinol and febuxostat, then you basically decrease the uric acid levels, which means you decrease the uric acid deposition, right? The uric acid crystal deposition into their joints, so therefore you would decrease the overall, pain and inflammation that they go through. All right. Another drug that I think, so those are the drugs that are involved in the purine salvage pathway so far, right? So we have allopurinol, febuxostat, and we have pentostatin and cladribine, right? Another drug we should talk about is called probenecid, and this is just like, it's not going to be that long, right? The mechanism of action, but basically, remember, we have in our kidneys, we have a section called the proximal convoluted tubule, right? Proximal convoluted tubule. So if we look at that more closely, let's say this is our tubular cell, right? In the proximal convoluted tubule, let's say this is our lumen on this side, and then let's say we have our red bloods, or we have our blood vessel on this side, right? So our peritubular capillaries, for example. Okay, so our lumen is on this side, and we have our, let's say, our red bloods, or our blood vessel on this side, right? So our peritubular capillaries, and this is going to be our proximal tubular cell, proximal convoluted tubular cell. My bad. All right, so let's say you have uric acid, right, in your filtrate on this side. So let's say you have uric acid, and we'll go over like renal physiology and all that in a separate playlist. But for now, let's just understand this, that let's say we have uric acid in our filtrate, right, that's going down our lumen. On the side of the lumen of this tubular cell, we have a transporter, okay? This transporter is called, it's abbreviated URAT1, okay? Basically, it's a urate transporter type 1, okay? Urate transporter type 1. And what this transporter does is it takes uric acid and transports it, right? And ultimately uric acid can get transported back into the blood, or reabsorbed into the blood, I should say, right? So you're going to have uric acid here. But what probenecid does is it comes here to this URAT1 and inhibits it, okay? So probenecid, let's write it down here, probenecid, this drug, right, comes here and basically inhibits this transporter, which means you will not reabsorb now uric acid back into the blood. Instead, you will pee this uric acid out, okay? So this will end up in your urine. So that's good because, again, for people who have elevated uric acid levels, right, this drug would help excrete more amounts of uric acid, which would decrease your overall blood uric acid levels, right? So that's probenecid. And then lastly, right, a drug I want to talk about is, or an enzyme first that I want to mention is, when you produce uric acid, you can also, not us, okay, not in humans, because of this enzyme. There's an enzyme that can convert uric acid into allantoin, okay? And this enzyme is called urate oxidase, okay? But urate oxidase is not in humans, okay? It's not present in humans. Specifically, you can find urate oxidase in a specific fungus species, okay? And that fungus species is called Aspergillus flavus, all right? So in Aspergillus flavus species, you can find urate oxidase. But what we can do is we can make a recombinant form of urate oxidase called rasburicase. Okay, so if we're just focusing on the basics of, recombinant DNA and all that, because we can go over that in future videos. But right now, let's just focus on the basics. So it's not going to be each and every step, right? We'll skip steps, but we'll mention the basics of this recombinant DNA process, right? And then that's how we can understand, okay, so that's how rasburicase is made. Just the basics of it. So, let's say this is the urate oxidase gene, right, in our Aspergillus flavus fungus species. So what we can do is we can take some restriction enzymes, right, which are basically just enzymes where, it's specific to the gene you want to cut. So I can take restriction enzymes and it's specific to the gene, right? So then it'll cut out that gene, and then we can take that gene and take a plasmid, okay, and insert this gene. Of course, we'll need other enzymes like DNA ligase, for example, right? But again, we're not mentioning each and every step of this recombinant DNA process, right? Once we insert this urate oxidase gene now, that again we got from Aspergillus flavus, the fungus species where urate oxidase gene is found, we can take that gene, cut it out with restriction enzymes, and then insert this gene into a plasmid. So this becomes a vector now, okay? And we can take this plasmid and through the process of transformation, which is basically bacteria taking in DNA from the outside, right? Through the process of transformation, we can take this plasmid and put it into the bacteria. Then again, we can let the bacteria replicate or proliferate. And then we can take out this urate oxidase. We can take out urate oxidase, the enzymes, right, from this bacteria, because now, the urate oxidase gene has replicated, and there's now many bacteria cells with this urate oxidase gene, so we can take out the enzyme, right, which is urate oxidase, because that's the enzyme that the gene codes for, right, urate oxidase, the enzyme protein. Take that protein out, or urate oxidase out, and we can undergo certain purification processes and get our we can use this medically now, right, which is called rasburicase, which is basically the recombinant form of what? Urate oxidase, right? Urate oxidase. So we didn't have urate oxidase, right, in our cells, because it's not present in humans. It was present in Aspergillus flavus, the fungus species, right, or the fungus. But then we took that and underwent some of the recombinant DNA processes, and then got rasburicase, which is now medically usable. Now we can use this because it's literally the same function as uric oxidase, right? And then what does rasburicase do? Go down here. Rasburicase will do the same thing as uric oxidase because rasburicase is the recombinant form of uric oxidase, and rasburicase will help convert uric acid into allantoin. Okay? But why is that useful? Why even do that? Again, allantoin, if you compare allantoin to uric acid, so uric acid, if you compare that to allantoin, uric acid is much less soluble in water than allantoin. So what we should say is that allantoin, right? Allantoin is much more soluble. So allantoin is this means much more soluble in water than uric acid. Okay, so that means it's going to be better in order to get rid or excrete through urine, right? Allantoin will be easier to excrete through urine than uric acid will be. So that's why it'll be cool if we can convert uric acid into allantoin, right? Especially in cases where people have elevated uric acid levels. And something else that, I was trying to look at, there's some, like if you're treating chemotherapy, right? Or if you're undergoing chemotherapy, that can, destroy tumor cells pretty quickly. And so that can lead to many things, right? Like let's say these are your tumor cells, okay? And chemotherapy leads to the lysis of these tumor cells, right? And what do we know that's inside of cells, right? Potassium. So if you have a lysis of a bunch of tumor cells, you have a bunch of potassium that can leak out into the blood. So there's something called tumor lysis syndrome. All right? And tumor lysis syndrome is basically when, again, you're undergoing chemotherapy, let's say, and a bunch of tumor cells start lysing or getting destroyed, and so that can lead to a sudden increase in potassium, since most of your potassium is inside your cells, right? So that can lead to, let's say, for example, hyperkalemia, which is elevated blood potassium levels. And also, your cells, usually you're maintaining a low calcium levels inside your cytoplasm, right? And there's more calcium outside your cells than in your cytoplasm. There's more calcium outside your cytoplasm, right, when compared to inside your cytoplasm. But if you start lysing all these tumor cells, then the calcium can start leaking in from the blood, right? So if your blood vessel's right here, you can have calcium coming from your blood and leaking into your cells. So we can have some things like hypocalcemia, right? If we're talking about all these tumor cells that are suddenly lysing because of chemotherapy, calcium can start leaking into these cells, which can cause hypocalcemia, low blood calcium, and because, again, the calcium in the blood can start leaking into all these cells that are lysing, right? And so that could lead to low calcium levels. We could have high potassium levels. But this is just a little background to tumor lysis syndrome. What's the main thing we have to focus on, right, is that what's inside your cells, genetic material, right? So that means that we can also have a bunch of DNA, right, that also gets released out from the cell. And we know that more DNA release means we can metabolize that DNA more, break it down more, right? So if we break down DNA, what is DNA? What's one of the things that DNA is made of, right? Purines. So if we break down and metabolize the DNA that's present inside your cells, because we're destroying it now, so we can break down that DNA, and that can lead to purines, more and more purines, right? And remember, we've been going through the Purine Salvage pathway. What can this lead to? This can lead to increased amount of uric acid, right? At the end of the Purine Salvage pathway, remember, we can have a bunch of uric acid because ultimately these purines can, has the possibility to get converted to hypoxanthine, then xanthine, and then ultimately to uric acid. So that's the significance of using, for example, right, like rasburicase, for example, or any drug that reduces your uric acid levels. That's the significance of it, like a clinical significance, because, if you're doing chemotherapy, right, if your cells are undergoing chemotherapy, that can lead to tumor lysis syndrome. Not only can that lead to, a mismatch of your ions, right? So for example, we can, like we mentioned, you can have a bunch of potassium leaking out, bunch of calcium from your blood leaking into your cells, and then you can also have, because your cells contain a lot of genetic material, that can get metabolized into purines, right, especially your DNA, and then that can lead to a bunch of uric acid, right, increase uric acid levels, and that's not good because this can also way too many uric acid levels or way too high uric acid levels, right, can, like, we know that uric acid crystals can deposit into your, tubular cells of your kidneys, right? So increased uric acid crystals isn't good for your kidneys, right? Significantly elevated uric acid crystals, not only can it deposit into your joints, right, and cause gout, but it's also not good for your kidneys because it can cause kidney damage too if it's very high uric acid levels, right? And that can deposit, specifically the crystals can deposit into your tubular cells of your kidneys and may cause kidney damage. So it's not good for your kidneys either, right? Or it's not good for your joints because you can get gout, which can lead to a lot of pain and inflammation, right, and whatnot. So that's why drugs that basically, like rasburicase, either that will convert uric acid to allantoin, making it more water soluble, so you can excrete it better, or there are drugs like, allopurinol, for example, that will inhibit xanthine oxidase, so you can't make much uric acid, which will also help decrease uric acid levels in cases of hyperuricemia. All right, so I know that was a long video. That's my bad, but now we understand the drugs that we can use in the Purine Salvage pathway. And so that's it for this video. In the next video, we'll talk about, adenosine deaminase deficiencies and more diseases relating to the Purine Salvage pathway. All right, so I hope to see you guys there, and thanks for watching.